Software engineering is a deterministic field. We write lines of code and feed data into that code, expecting to get a certain answer. Computing is deterministic because humans developed it. We understand computers from top to bottom. The same cannot be said about biology. Matt Might is an associate professor with a PhD in computer science. When his son was diagnosed with an extremely rare illness, Matt was confronted with the uncertainties of human biology. In this episode, we discuss Matt's quest to solve the puzzle of his son's disease. Computer science meets genetics on this episode of Software Engineering Daily. If you're a fan of Software Engineering Daily, please tell us how to improve. Fill out our listener survey on our website or in our newsletter. There are links in both of those places. We would love to know what you think. We would love to know what you want to hear more of, what you want to hear less of. We listen to all of the feedback that we get, so please fill out the survey on softwareengineeringdaily.com or in our newsletter. After a quick word from our sponsor, we will get to this episode of Software Engineering Daily, Genetic Sequencing and Computer Science with Matt Might. As a software engineer, you have tremendous leverage in the employment market. Every company needs software engineers, and these companies are all competing for you, the engineer, a scarce resource. But as an individual engineer, you don't have much insight into this incredibly opaque market. That's where Hired.com comes in. Hired.com assigns you a talent advocate who works to understand your needs and works to find you the perfect job. Go to hired.com/SEDaily to try out the platform today and get a $2,000 bonus if you find a job. The companies on Hired compete for you, the engineer, and they make bids to win your talent. These are companies like Facebook and Uber, companies that we know are on the cutting edge of engineering because they've come on Software Engineering Daily to talk about their tech stack. I've used Hired personally, and I've experienced firsthand the ease of finding a well-paid, enjoyable job. This was way better than replying to LinkedIn messages from uninformed recruiters, and it was also certainly better than using a job search engine. To find a great job and support Software Engineering Daily, go to hired.com/SEDaily and get a $2,000 bonus upon finding a job. Check it out. It would support the show. Now let's get on with the show. Matt Might is an associate professor with a PhD in computer science. He's currently on sabbatical as a visiting professor at Harvard Medical School. Matt, welcome to Software Engineering Daily.
Thanks. Good to be here.
Scientists and engineers are always experimenting, and experimenting often means performing tests and assessing the data across a large population of sample cases. But you've talked about how to approach a scientific situation when the experimental population has a sample size of 1. And this idea became important to you because your son was diagnosed with an extremely rare disease. Before the birth of your son, had you done much thinking about how to assess scientific situations with an extremely low sample size?
Well, that's a great question. And prior to my son, not really per se, although in some sense I did a little bit of that already in computer science. So my native research in computer science was actually in areas like static analysis and program optimization, largely applied to functional programming languages. So languages like Haskell or Racket. And believe it or not, it was difficult at times to find large code bases to apply our techniques to. So we often had to infer that they would scale if they could scale on larger code bases, from looking at how they behaved on smaller program fragments. So instead of looking at data in the aggregate, we were looking at how well it performed very specifically and exactly what it was able to infer about a particular program. Before that, it's certainly not in a medical context, certainly not in a broader scientific context. I had sort of a little bit of experience with it in my native research in computer science. And in fact, you say it was very rare, but to be clear, it was so rare that he was literally the first case ever discovered. So that's about as rare as it gets. Now, we've since discovered more cases of it around the world, but it's still one of the rarest diseases on the planet. Certainly not big enough to ever have the kind of sample sizes that you would need to draw the levels of statistical confidence that a regulatory agency like FDA would want if they were to approve a drug for it.
After your son's birth, there was a 4-year period where your son was suffering, and it was suffering from a mysterious condition, and there was nothing that doctors or scientists could do to provide information. Did this experience make you question any of your fundamental beliefs about the effectiveness of science?
Well, it certainly had me questioning beliefs in general. Though ultimately, the entire process has been a confirmation about really the importance of science. Where a lot of my beliefs were questioned were on the modern medical system, and how in many ways what I came to realize is that modern medicine is fantastic at treating the average. It's set up to treat the general population. And if you happen to be one of these rare cases, it's really not set up for you. And yet, it will almost act as if it is. I mean, we tend to place a tremendous amount of trust in physicians, and most of the time, rightly so. But there's a problem when you enter into areas of disease this rare, where oftentimes the medical system won't admit the limitations of its own knowledge, and doesn't really have a path for patients like my son to take. And it's not like my son is the only one. There's a lot of rare and undiagnosed patients out there. And I've heard now that from places like the Undiagnosed Diseases Network that the average length of an undiagnosed odyssey where you're looking for an answer is 7 years. So when you get into rare disease, it can take a very long time to finally figure out what exactly you have. So I certainly question my beliefs and the trust I placed in modern medicine, at least in the way it's delivered currently. I think there's a lot of room for improvement there. But in science itself, I know you're always dealing with the threat of the unknown. That's really what science does. We stand out there at the boundary and we shovel away, we push, and try to figure out more stuff. And there's always the risk that you're going to end up being the very first case of some brand new disorder. It has to happen to somebody, and it happened to happen to my son. To me, actually, to watch that process of discovery take place was really inspiring.
So when doctors figured out that your son was an N of 1, as you call it, a unique case, they said things like, this case is not actionable. And I think this gets to what you're saying about fitting outside of the average, being an outlier when you're engaging in treatment. So what was the basis for the claim of the doctors saying that this case was not actionable?
Yeah, so actionability is actually, it's one of these terms that's somewhat well defined within medicine. And essentially, if there's no pill you can take or no procedure available, then it's considered not actionable. If there's nothing that medicine can offer you in the advent of a diagnosis, they call it a not actionable diagnosis, or sometimes they'll say there's no clinical utility in the diagnosis because there's nothing that the medical establishment itself can do for you at the time. I take strong exception to that at this point because I think that science itself should be included as an action that you can take in the event of a diagnosis.
So you disagreed with the idea of this case not being actionable. And you've said that in the absence of any traditional medication, which is an example of this type of situation, they didn't have a traditional medical solution. So in the absence of any traditional medication, science becomes the medicine. What do you mean by this? Science becomes the medicine.
So by that, you get to begin the process of understanding the disease, if it's not understood as it was in this case. And if it is somewhat understood, then you can begin the process of actually doing the bench science necessary to develop drugs and treatments. So for example, with a brand new disease, the first thing we did was, there's literally nothing known about this. So we have to do some basic cell biology to start understanding what we're dealing with. And more in particular, what we're not dealing with. There was a lot of hypotheses about what this disorder could be doing and what it might be doing. And so we had to start testing those hypotheses. So we began collaborating with a glycobiologist out at Stanford Burnham Research Institute in San Diego named Hudson Freeze. And, he allowed us to sort of, again, start not only testing this hypothesis, but putting more hypotheses out there to say, we think this could be a mechanism in this or this, or maybe this isn't. So the first step, for us, and I think for many of these ultra rare diseases, is to start to understand, to unlock the mechanism of harm. And the mechanism of harm, it's really a chain that begins with the initial genetic insult and ends with the high-level symptoms you see in the patient. And in between, that initial genetic problem and those symptoms, there's a long chain of causes. Like, in my son's case, the genetic change causes an accumulation of glycoproteins. And it actually causes a very specific kind of accumulation of glycoproteins, we now know, and that the kind of accumulation does appear to matter. So, and realizing that the kind of accumulation matters has allowed us to develop therapeutic strategies based on that fact. So, at first, I would say science is doing the basic cell biology, doing very basic science, just to get a sense of what's actually happening, and over time, you'll start to see opportunities emerge that could paint the way towards therapies. And there's other more general things you can do too. So you can do classic drug development, but miniaturized for an individual patient or for a very small patient population. And the first stage of that is really to look for what are called phenotypic modifiers, or to find drug targets, which in this case would be mostly genes, that when you interact with them in some way, they modify the disorder. And you can either make it better or worse. So if you find a gene, for example, that when shut down happens to make the disorder better, then you have a suppressor target. So you want to try to find an inhibitor or an antagonist or whatever the case may be for that particular gene to try to knock its activity down, under the understanding that that will actually help the disorder. Conversely, you might find that some genes, when overactivated, actually help the disorder. And so in those cases, you want to find compounds that will overactivate those genes. And there's a number of techniques you can do to even identify what these other genes might be. So there's several kinds of modifier screens you can do when you've got a brand new disease. And so that's another kind of science you can do right off the bat when you discover a brand new disorder. And we really did both in parallel. We started looking for modifiers, and we started trying to understand the basic cell biology right from the get-go.
So when it comes to the modern experiment process of trying to find some genetic clues or some genes to target, what is at the cutting edge of this type of process? Like, as you've engaged with scientists and researchers and engineers about this problem, what kinds of technical improvements, software, hardware, are you seeing on this front?
Well, that's a great question. So I think it's amazing how fast things have changed in just the past few years. Even the development of CRISPR, I think, is definitely a game changer when it comes to working on the biological side. So CRISPR is a fantastic gene editing technology, and the way I would present that to a software engineering person is, imagine that you have Emacs for your genome. That's CRISPR. I mean, you can go in and make whatever modification you want to an individual cell. It doesn't work in people yet, but it does work fantastic for creating model organisms where you want to mock up a disease in a model organism. And model organisms, I think, are still one of the best ways to do a modifier screen. I think I can mention a couple ways to do this with a computer, but in many ways, nature is still the fastest computer we have when it comes to studying disease. So if you want to find a gene that might have an effect on the disorder you care about, what you can do, and there's several ways to do this, but one of them is, first, you create a model organism like a worm or a fly that has the genetic disorder you care about. And then you can either systematically start knocking out fractions of that model organism's genome to see if suddenly it gets better. So if you knock out some fraction of like a fly's genome that has a particular genetic disorder and it gets better instead of worse, then you form a hypothesis and you say, well, maybe there's a gene in that region that actually, if we take it out, makes the disorder better. And you can try to zero in on what that gene is. So there are existing libraries of flies which have basically preloaded with large fractions of their genome knocked out in a defined way. So you can cross your fly that has a genetic disorder or as a carrier for it with these, and end up effectively screening every fragment of the genome to see if there might be a modifier there. And you can do similar things in zebrafish or in worms. Again, depending on what model organism you have, there might be other ways of doing this. The other way to do it is to create an organism with a genetic disorder and then blast it with mutagenic agents. And again, you're not making just one organism, obviously you're making many. And if some of these organisms start to get better after they've been exposed to something that's mutating their genome, then you say, oh, well, let's go backwards, let's sequence them and find out what we did to it that made it better. So there's a couple ways you can go about looking for modifiers at the bench. Computationally, there's actually a lot you can do now. And again, this is all stuff that's only becoming possible quite recently with the advent of cheaply available genomes. So one project that we're working on here at the University of Utah is going to combine all of the genomic data we have on all the known NGLY1 patients to date. And we're going to score each patient according to their essentially the severity of their presentation with the disorder. And there's software that can actually then look for the genes that are modifying the disorder that way. Because no patient is identical to any other patient for almost any disorder. And when you get a spectrum of severity in a disorder like we do with my sons, then you can reasonably assume that it's due to either environment or other genetic factors. And so it's hard to account for environment, but certainly if there are other genetic factors influencing a disorder, you can try to hunt those down as a giant data science experiment. And so that's another interesting thing to point out is that even though we have a very small amount of patients, we're not data poor. We have a tremendously large amount of data per patient at this point.
So this process of, it seems almost kind of like a random walk, and then you look for something that works, and then you work backwards to determine why it works. Does this ever feel like a process that is more non-deterministic than it fundamentally needs to be?
That's a great question. So you're right. There's a tremendous amount of stochasticism, if you will, in the process. Not quite totally non-deterministic, but you certainly you're going to get drawn in a certain way based on.
Or it's like NP complete. Like it's not non-deterministic, but like it's so big that we couldn't explore it in a billion lifetimes.
Well, that's absolutely true. It's like when you get into drug development, people will talk about what they call chemical space. The space of all possible molecules that could be a drug, and I forget the number for how many there could be, but it's just enormous. I mean, the number of ways that you can rearrange, simple organic compounds is just staggering. It's one of these very, very large numbers, and so we know in advance that humanity is never going to be able to search out all of chemical space in a systematic fashion. We have to do some kind of heuristic search or probabilistic search or of this space as we attempt to map diseases to drugs. And just I don't know if it will ever be totally deterministic. I think we're always going to have to rely on an element of to some degree random exploration, but hopefully guided random exploration, so that we're trying to move in a direction that our intuition tells us might be a reasonable place to go.
So I'm not an expert on deep learning, but we've done some shows about deep learning, and one of the characteristics, as I understand about deep learning, I could be wrong, is that it's this process of oftentimes you're searching for a solution, you have this objective function you're trying to optimize, but oftentimes you end up with a solution, you don't exactly know why your solution works, all you know is that it works. This is a fascinating, extremely powerful set of tools, but I'm curious if you understand much about the space and have any idea how it could be sort of transitioned from the bit world to the atom world, to use the buzzword phraseology of it.
Sure. So I'm not an expert in machine learning and not in deep learning. I understand it about as well as you do probably.
Does that sound accurate, what I said?
Yeah, it sounds accurate from my understanding. And once upon a time back as an undergrad, I played with neural networks and things like that. So I have the general idea of what's going on here. And yet there's this. I also understand the complaint that when it gives you back an answer, you can recognize that it's right or that it's good, but it's very hard for it to explain why that was a reasonable answer. And so I do have a graduate student working on using machine learning techniques, not even deep learning yet, to understand to build classifiers for glycogenes and things like this. And even before you get to deep learning, it can be sometimes difficult to understand what the model is telling you. Like when it synthesizes, a set of weights to do classification, and you try to look at those weights and make sense of how it learned what it learned. Even even that can be challenging. We were trying to do tissue type identification by looking at levels of gene expression, and eventually we figured out that the way it had taught itself to recognize tissues was to recognize what tissues it wasn't. If that makes any sense, because it was just all these large negative weights to penalize most tissue types. So that is, as opposed to giving very positive scores to markers of what the tissue actually was. So that was an interesting finding when we looked at that. But there's actually there's an analog to drug development. You would be maybe shocked to learn that there's a lot of drugs out there where we have no idea how they do what they do. Or very little idea of how they do what they do, and that's because there's sort of two ways to go about this. You can be very principled and try to rationally design a drug to do exactly what you want once you've identified a target, or you can do broad compound screens where you can test millions of compounds against some model or some cell with respect to a test that tells you whether or not it might work, and then you get some hits and you may have literally no idea why it does what it does. All you know is that it does the thing you wanted it to do. The generate and test approach is very popular in modern drug development, where you literally just blast a disease with millions of compounds in dishes, hoping that something makes a difference in a recognizable way.
I've experienced this in the domain of psychiatry, where I was in a psychiatrist's office, discussing medication. And his way of explaining how the medication worked was drawing, on the x-axis was time, and on the y-axis was mood, as if these are this is an abstraction that actually makes any sort of scientific sense. And then just drawing the graph of here's how your behavior looks before the drug, and here's how your behavior looks after the drug. And that's the best they could do. That's not me criticizing science. That's literally the best that they could do right now.
Yeah, it is. I mean, so actually you bring up, there's certain areas of drug development which are just enormously complicated on top of the already big challenges they have. And one of them is psychiatric drugs, because suppose you do want to do a lot of testing. What is your test? What if you're looking at cells, or what if you're looking at lab mice in the early days? How do you know if it's having any kind of psychiatric effect at all? Measuring the effectiveness of drugs is incredibly difficult to do in many spaces, and especially so when you get into drugs that are supposed to make a difference for our mind.
Right. So I want to talk more about the story of your son. So after your son was diagnosed, you kind of took this matter into your own hands because it was not actionable according to the doctors or the researchers. And you decided to talk to the top researchers in the field, and you resolved to research and work on finding a treatment for your son. And you reasoned that there were, probably 500 other people living with this rare condition that your son had. And you realized that you couldn't solve this problem alone, so you made a blog post that was designed to go viral, and you ended up finding a lot of people because of that blog post. And once you had a group of 35 people identified, you could start to do some more natural experiments. You started to gather biomarkers. So give me an idea of what this process was like after you started to have some people, you started to be able to gather some data. When did you get into the high throughput drug screening process?
Well, that's a great question. So we're still on the threshold of high throughput drug screening. We have an assay that can detect whether the enzymatic activity of the missing enzyme is present again. But we think it's highly unlikely that any molecule would somehow magically bring back that activity. So what we're looking at now are we're looking at sort of higher level mechanistic assays to see if we can influence, say, the seizure phenotype. And that looks like it'll be a much better basis for doing something high throughput, where we would scale to millions of molecules. And in lieu of doing high throughput, we've been doing very targeted drug screens instead, where we have a proposed way it's going to influence the mechanism or some mechanism of harm, and then we can check to see if it actually does that. But yeah, once we got patients, it taught us a lot. So first of all, it taught us that everybody presents with this disorder slightly differently. There are no two patients that are exactly alike. I mean, there is definitely a core set of symptoms of this disorder that all patients share in common. But when it was just my son, we didn't know what was really the core of the disorder and what was unique to him because of his own genetic variation. And right off the bat, it's important to figure out what's the core of a disorder, because if you spend your time treating stuff that's on the periphery, then at best you're going to get sort of skin deep in actually treating the disorder. Whereas if you focus on the stuff that's fundamental to the disorder, then you're starting to influence deeper levels in this chain of mechanism. And it's really important for drug development to understand what that core is, so that you can so that you can address it as deeply as possible. But one thing that happened also from getting a lot of patients is we started to realize that, yes, there is an actual spectrum of patients, and some had different experiences medically and also in terms of their genetic background. So one of the patients, for example, had a liver transplant because in addition to having this ultra-rare disease, he also had liver cancer. And he went from having essentially around-the-clock seizures before the transplant to not a single seizure afterward. So obviously that got everybody's attention. And the more we've pushed on this, the more we've started to realize that, because the initial thought was, oh, well, maybe everyone needs a new liver. Because, it's possible to do that for all the patients, but it's a very dangerous thing to do. So you don't want to do it unless you really have to. So we said, well, let's see if we can understand why this might happen. And then we started to realize that it might not actually be the liver transplant at all. It could actually be the particular immunosuppressant that this child was on. It turns out that the immunosuppressant he's on upregulates a process called autophagy, which is an alternate way of clearing out some kinds of cellular waste. And since this disorder is fundamentally a breakdown of a pathway that handles cellular waste, it looks like this is a possibly way to actually compensate for that. Now, the downside is the immunosuppressant he's on, you would never, it's not as awful as a liver transplant, but you certainly would not want to go on it unless you had a really good reason to, because you're going to get sick all the time. It's not a fun lifestyle once you go onto it. So we're looking for other compounds that could also induce this same process of autophagy. And we're actually trying to test those in model systems to see if these less harsh compounds could provide the same degree of benefit. Now, if they can't, then maybe there's something else about this immunosuppressant that's doing it too, or maybe it really is the liver transplant. But at the moment, we think that this patient has sort of shown us a possible way of actually treating the disorder through this alternate process called autophagy. And then we also have some patients that are very high functioning relative to the rest of the patient population. Again, there's no question what disorder they have, but instead of being, say, 6 to 9 months old developmentally, they're out, mainstreamed in school. They can, walk, talk, sing, dance, use iPads. It's very, very different from the rest of the patient population. So, of course, we're looking very carefully at their genetics to see, well, what modifiers did they just happen to get by a stroke of luck that changed this disorder so radically for them? Because if there's a process that's upregulated or downregulated in them that's actually influencing the disorder, maybe we can find pharmaceuticals that will manipulate those pathways in all the patients. So that's the kind of stuff that happens once you get lots of patients.
Your company has important projects that need to get done. The iOS app needs to be rewritten for Android. The database needs to be migrated. Your continuous deployment system needs to be built. The website needs a complete redesign, but you don't have enough software engineers and designers to get all this work done. TopTow is here to save you. TopTow gives you exclusive access to the top 3% of freelance talent. Software engineers and designers, from Python to PHP. TopTow has the freelance talent you need to get your projects finished on time with top quality. In the past, we had to worry about flaky freelancers with poor communication skills, unreliable internet connections, subpar technical skills, and so on. TopTow screens for these kinds of things and only works with seasoned professionals with tremendous problem-solving skills, personality, and drive. Here's how it works. TopTow's internal team of senior engineers will work with you to understand your project scope and your talent needs. and they will custom match you with just a few hand-picked candidates. This means that whenever you need to add top-shelf talent for a critical project, you can be connected with pre-screened engineers who are hand-picked for your needs. And the results are impressive. TopTow clients conduct just 1.7 interviews for every hire that they make. All you need is to come ready with some decent technical specifications of your project, and TopTow's team of engineers will take care of you from there. If you are looking to add critical talent fast and you need a source you can trust, go to toptow.com/SEDaily. You can also send me an email directly at softwareengineeringdaily@gmail.com, and I will personally introduce you to the team at TopTow so that you can learn more. We live in unique times. The nature of work is changing, and more industry-leading companies from Airbnb to JP Morgan are realizing the benefits of scaling quickly and staying flexible by working with elite freelancers. So if you're short on resources for your projects, check out toptow.com/SEDaily. Thanks to TopTow for sponsoring the show. Now let's get on with this episode. Talked about these two types of screening. There's high throughput drug screening and targeted screening. Could you contrast these two?
Yeah, so high throughput screening is agnostic as to sort of the way something's going to work. You're just going to say, I'm going to try thousands or even millions of different small molecules in the hopes that one of these actually makes a difference. So what you do is you come up with some test that recognizes when something's been improved. So usually this is the level of cells when you're talking about millions of compounds. You will have an assay that literally, in our case, it will glow fluorescent green if you have found a compound that makes a difference for the disorder and in terms of actually restoring deglycosylation activity. So, what we could do is we could, drop millions of compounds into millions of little dishes and check to see if any of them turn bright green. And if one of them turns bright green, we say, aha, that compound made a difference. And the thing is, we're not going to know why. We're going to have literally no clue why that did that. We could do some investigation and maybe find out, or maybe we wouldn't. When you do a targeted screen, you may be screening, dozens to hundreds of compounds in a particular class because you have a theory of what's going on with the disorder. For example, if we do think that autophagy is actually causing this disorder to get better, then what we would do is select compounds that are believed to increase autophagy and test those on cells with some assay for autophagy-related activity. And if we see a difference there, then we say, okay, well, it looks like we found autophagy-inducing agents, and if we have other assays for checking other mechanisms of harm, like for example seizures, then we could do a secondary, even more limited screen with the ones that passed the first screen. So you can do layers of targeted screening when you've got a specific hypothesis about why a disorder behaves the way it does.
So you've also done what you call ad hoc crowd screening by communicating on platforms like Twitter and Reddit and Hacker News. How have you been able to leverage these social communities to get research done?
It's been phenomenal to watch this happen. So some of our most interesting compound suggestions and suggestions about potential mechanism of harm have actually come from social media. They've come from places like Reddit or Twitter. So early on, when the blog post was going viral, a postdoc who worked on cancer made some comments, like some just incredibly insightful comments to the point where I'm like, okay, this guy clearly knows what he's talking about on Hacker News, I think it was Reddit or Hacker News. I can't remember which one it was, but I followed up with him. He's like, yeah, I'm a cancer researcher, and this isn't cancer, but you're trying to deal with these misfolded proteins, and I know a lot about molecules that could potentially chaperone this protein and maybe help stabilize it. So he made some very insightful recommendations on the forum, and then I gut checked those with the scientific team, and they said, "Oh yeah, that's actually a fantastic idea. We should try this out." And so over the course of the past several months, we've actually reached the point now where we're starting to test this Redditor's hypotheses in the lab with real compounds. Again, all based on suggestions that he had been able to make. And then over Twitter, when we were discussing this, my wife actually made contact with a postdoc in Texas who said, I'm actually working on cystic fibrosis, but I think that the same compound I'm developing for cystic fibrosis will also work for your son's disorder. In fact, it will work specifically for your son's mutation. It's a mutation-specific drug, so it could work for a variety of genetic disorders, not just cystic fibrosis or my son's. He said, "Would you mind sending me some of your son's cells?" So we, got him in touch with Dr. Freeze out in San Diego and said, "Can you ship him some cells?" He did, ran some tests, and sure enough, it looked like he was actually able to restore a very small amount of protein expression at first. To what levels that might actually be therapeutic, and then after some optimization, was able to restore large amounts of the protein missing in my son. So that's actually turned into a startup in the process of these discussions. In between when we met on Twitter and now, and they're looking for early funding and might even have an IND approval within 18 months. Or IND means it gives them the ability to start looking at doing clinical trials for this drug. So that's incredibly exciting to think that a Twitter conversation led to cells shipped across the country, which led to 2 rounds of testing and incremental improvement of this compound, which may ultimately lead to a clinical trial.
There seems to be a transition happening within the world of science, or at least I hope there's a transition happening to an increased level of communication, because I feel like there are some sectors of science that are like heavy research, basic science, where there are these silos that exist where people are doing research on their own. They want to be totally isolated from other people because they don't want to be scooped. They don't want to communicate with others aggressively. They don't want to use Facebook or social media or whatever because they might be scooped. And I see this as totally crazy because if you think of like the scientific population as a whole, if you risk getting scooped, that means that somebody else in the world is doing duplicate work, and you should prevent them from doing duplicate work or stop doing duplicate work yourself. And so all that is to say is, like, do you think there is an allergy to social media and sharing to the same degree that the open source community of software has embraced it?
It's very different. You're absolutely right that particularly in the biological sciences, that there's a lot of siloization going on, and certainly even in computer science as well. There are some silos there as well. Although I think we're much better about sharing code and data than we are in the biological community. And there's definitely a sense. In fact, there was a recent editorial in the New England Journal of Medicine by some of the editors that said that those who would use other people's data if it were released openly to produce their own results are research parasites. That was the mentality. Literally, they use the term research parasite.
It's so zero sum.
Yes, it's a very zero sum sense, and part of that is it's an artifact of the way that we have set up the recognition of credit in science. So we have one unit, to maybe 2 units of recognition. So we have papers and we have grants, and even within a paper, it starts to get fine-grained because there's like who's first author, who's the corresponding author, we need to make it more granular than an entire paper, and we have to have recognized ways of saying, look, this person has in fact made a meaningful contribution, but maybe it didn't turn out as a paper. Maybe it turned out as other people got papers because they built a tool or a platform that enabled them to do that. We're just now starting to recognize artifacts in computer science. So there's new guidelines from the CRA saying that when you're reviewing people for tenure, in addition to reviewing the papers they produced, also consider the software artifacts that they used and how widely used those software artifacts are. So, like, if you produce a software tool that lots of other people are clearly benefiting from, then that should count towards your scientific record when you're coming up for tenure. And but it's going to take a while. That requires a cultural shift. So I don't know what the exact solution is here, but we do have to find alternate ways of giving credit to people for what they've done, and for the ideas that they have. I think we'd be in a much better world if we incentivize all scientists to just dump whatever their hypotheses are out there right now, so that if 2 people have the same idea or they're doing the same thing, they can collaborate right off the bat, and then split the credit for that.
So I want to draw towards a conclusion of this conversation. When I think about your experience, I think about the fact that as a human in this constantly changing, unique set of circumstances that is the experience of being a human, I think we're all in this N of one situation. Like we all have our own unique genotype. But the way that we're taught to think is in terms of the average circumstances, like you ended up running up against in the non-actionability of your clinical scenario. Do you think there's a philosophical shift that individuals should make, maybe not even just because of not just thinking about like their medical circumstances, but thinking about their lives as a whole? Should people compulsively think of themselves as an N of one, as an outlier, as something that is like an entity that is unique and not bundled into the average population?
Absolutely. No, I totally agree that regardless of your circumstances, we are all very unique. Even identical twins have different experiences, even if they share most of the same genes. So I think we all have to recognize that, life is the ultimate N of one experiment. It really is, and you've got to treat it that way. And honestly, there's no fun being average. I think we should all exploit the fact that we can be outliers if we want to.
Yeah, and the writer Tim Ferriss encourages his readers to approach this compulsively, and he says that it's helpful to perform N=1 experiments on yourself. And the axis that he suggests using to expand the sample size is time. So like you can test A on yourself one day and then test B on yourself the next day. Do you think this is a good approach to life, to like try to do A/B tests across time on a personal basis?
Absolutely. I mean, I'm sort of a productivity nerd, and I run experiments like this all the time, where I'll try something out and see if it makes a difference or not. For the most part, the judgments you make about this are going to be, I think, pretty valid, particularly if there's a large effect. The only issue with using time in yourself is that you can't be your same self 2 times in a row. From one week to the next, you're always going to have slightly different circumstances. But you know what? You can sort of control for that by repeating the experiment over and over again. And this is actually something that happens a lot in rare disease, where you're experimenting with individual medications where you'll go on and off and on and off to see if it's actually making a difference. We've certainly had to do that. I know plenty of other people that have had to do that. And I think you can generalize that concept to almost any activity in life.
If you get diagnosed with a condition, or if a listener is diagnosed with a condition that makes that listener a N of one case and is just running up against the non-actionability of the medical community, what are some actions that they can take to try to find a solution?
First I'll insert a blurb to say that I'm writing a blog post on this exact topic. So if you don't know what to do, then I think you do want to try to find others. So I already have a blog post that describes how you can go find others and repeat the same process I used for my son. In fact, I've helped many other families at this point do exactly that, using social media to find other patients. But in terms of getting science itself done, I think that requires identifying a collaborator you can work with to get you through those initial stages of education. So depending on what it is, you need to do a literature search. So I would head to PubMed or to one of these expertise-driven search engines that lets you search for expertise across a number of medical disciplines, so that you can find the most closely incentivized collaborator for you.
You've spent significant time with biologists and doctors and computer scientists and other cross-disciplinary people. What are the similarities and differences between these sorts of fields?
That's a fantastic question. So a lot of the differences are driven by the differences in the way we do our science. So when you're a computer scientist and you want to run an experiment, you write some code, and you run it, and generally you get an answer back pretty fast. That's not always true in computer science, but usually you get an answer back very quickly. In computer science, we're used to a cycle of extraordinarily rapid iteration, where we just try something, might be quick and dirty, we see what happens, we modify it, we run it again. So we can do dozens, maybe hundreds of experiments per day, depending on what it is we're trying to do. In biology, you may set up an experiment and let it run for 3 months or 6 months or longer. And if you got something wrong, well, guess what? You got to wait another 6 months. So this creates a different culture between the two fields. I mean, there's this very creative hacker-like let's iterate rapid culture in computer science, whereas in biology it's more of we have very scarce resources. We must spend a lot of time planning how to deploy them in the first place. And so you spend a lot of time using intuition and judging hypotheses, because there's so many hypotheses that you could test, so many potential experiments you could run as a biologist, but you're only going to get to run a handful of them, and you've got to pick the right experiments to run. And there's also an element of luck to that, because you ultimately don't know what's going to happen, but you've got to figure out in advance some way of trying to guess what might happen. Whereas computer science, you could reasonably, in most cases, try everything.
Do you have a unique piece of wisdom that you've learned along this journey of diagnostics and medicine, and just your experience with your son, a unique piece of wisdom that we might not hear from anyone else that you can share with the listeners?
I don't know. I don't know about unique. I've certainly learned a lot. I think, at the end of the day, so here's the way I'll put it. If I could rewind 8 years ago to when my son was born, and you had told me all the stuff that I would have to do from then till now, I think I would have just crumpled up into a ball, and I don't think I would have been able to do anything. And even now, when I try not to think about too much about what I have yet to what's left to do. I focus very much on answering one question every day, and that question is, what is the next thing I should do? What can I do today to make progress for tomorrow? Now, it is definitely sort of a greedy algorithm if you think about it, but breaking it down like that makes almost any problem conquerable, and it doesn't seem so overwhelming at the end. So I would say, I don't know how unique it is, but no matter what your task is, no matter how big it is, just focus on finding the next step and just do that.
That's a great place to close off. Matt, thanks for coming on to Software Engineering Daily. I'm a huge fan of your writing, and your story is very inspiring. So thanks for coming on.
Well, thanks. This has been a pleasure.